Chapter 13 deals with meiosis and sexual life cycles. So the main concepts that we're going to be talking about include the following. Offspring acquire genes from parents by inheriting chromosomes. Fertilization and meiosis alternate in sexual life cycles. Meiosis reduces the number of chromosome sets from diploid to haploid. And finally, genetic variation produced in sexual life cycles contributes to evolution. So you've probably all heard of the term heredity. Heredity is a transmission of traits from one generation to the next. Variation is demonstrated by the differences in appearance that offspring show from parents and siblings. Not everyone has the same eye color, for example. Genetics is a scientific study of heredity and variation. We have a whole course here on genetics. If you're a biology major, you'll be taking this probably in your junior year. A gene-specific position along a chromosome is called a locus. Now, genes are the units of heredity. They're made up of segments of DNA. This is what is inherited. This is what's passed down from parent to offspring. Now, the way these genes are passed down to the next generation are via reproductive cells. These are called gametes. These are sperm and egg. Sperm and egg, your gametes, are haploid. That means they have only a single set of chromosomes. So in humans, they have 23 chromosomes because one set of chromosomes is 23. We use the letter N as a variable to represent the number of chromosomes in a set. Somatic cells are all cells of the body except gametes and their precursors. So these are all the body cells. So your epithelial cells, for example, your muscle cells, any cell that isn't a sperm or egg is called a somatic cell. Now in those sets of 23 chromosomes that we have in gametes, each has 22 autosomes, or what you can call a normal chromosome. It also has one single sex chromosome. The sex chromosomes are X and Y. In an unfertilized egg, the sex chromosome is X. An adult female is XX. In a sperm cell, it could be either X or Y because an adult male is XY. If we want to see the sets of chromosomes in the human cells, we can do what's called a karyotype. A karyotype is an ordered display of the pairs of chromosomes from a cell. So usually in order from the largest to the smallest, and then they show the pair of sex chromosomes. The chromosomes in each pair are called homologous chromosomes, or homologs. Chromosomes in a homologous pair are the same length, they're the same shape, and they carry genes controlling the same inherited characters. However, they can have different alleles. They can have different versions, which means that maybe chromosome 1 from your dad has brown eyes, chromosome 1 from your mom has blue eyes. They both have eye color, but they have different versions. Human females have a homologous pair of X chromosomes. Human males have one X chromosome and one Y chromosome. Only a small part of these two are homologous with each other. Figure 13.3 is showing a karyotype. On the left-hand side, it's showing how certain microscopes you can equip with a camera so you can visualize what's on the slide on the computer screen. And on the right-hand side, it's showing you an actual karyotype. So you can see they pair chromosomes up, so your 2 number 1s, your 2 number 2s, all the way down to your 2 number 22s, and then your pair of sex chromosomes. So this individual would be a male because the pair of sex chromosomes are X and Y. Each pair of homologous chromosomes includes one chromosome from each parent. In a cell in which DNA synthesis has occurred, each chromosome is replicated. Each replicated chromosome consists of two identical sister chromatids. This is something we talked about last year. Figure 13.4 shows maternal chromosomes in red and paternal in blue. This cells from an organism with a diploid number of 6. Each of the 6 duplicated chromosomes consists of two sister chromatids associated closely along their lengths. Each homologous pair is composed of one chromosome from the maternal set and one from the paternal set. Each set is made up of 3 chromosomes in this example, long, medium, and short. Together, one maternal and one paternal chromatid and a pair of homologous chromosomes are called non-sister chromatids. So this terminology is important to get straight. So on the left-hand side, you see where it says sister chromatids of one duplicated chromosome. So initially say this was your father's chromosome number 1, well, a father's chromosome number 1, it duplicated. Now there's 2 chromatids for his number 1 chromosome. On the bottom right, it says pair of homologous chromosomes, one from each set. So let's say whatever organism we're looking at, this is chromosome number 2. So you have a pair of number 2 from your father, 2 chromatids, and a pair of 2 chromatids from your mother for number 2. If you take one of the chromatids from the mother and one from the father, these are called non-sister chromatids because one comes from each parent. A life cycle is a generation-to-generation sequence of stages in the reproductive history of an organism. In the human life cycle, fertilization is the union of gametes, a sperm and an egg. The fertilized egg is called a zygote. It has one set of chromosomes from each parent. It produces somatic cells by mitosis and develops into an adult. In asexual reproduction, a single individual passes all of its genes to offspring without the fusion of gametes. A clone is a group of genetically identical individuals from the same parent. So as a result of asexual reproduction, you get clones of the same initial individual. In sexual reproduction, 2 parents give rise to offspring that have a unique combination of genes that are inherited from 2 parents. They're going to differ from their parents and from their siblings unless they happen to be identical twins or triplets, etc. Figure 13.2 shows 2 examples of asexual reproduction. These are both multicellular organisms. On the left is a very simple animal called a hydra. And it reproduces by just having a part of it budding off. You see it says parents and then bud. A little part of it reproduces by mitosis and it just buds off. On the right-hand side is an example of redwood trees. All the trees in this circle of redwoods arose asexually from a single parent tree whose stump is in the center of the circle. So these are both examples of asexual reproduction. Now in humans, at sexual maturity, the ovaries and testes produce haploid gametes. So the ovaries produce eggs, the testes produce sperm. Gametes are the only type of human cells that are produced by a process called meiosis. Meiosis results in one set of chromosomes in each gamete. Fertilization and meiosis alternate in sexual life cycles to maintain chromosome number. This slide, figure 13.5, is showing the human life cycle. So if we start with the 2 humans on the bottom left, humans are diploid, so they have 2 sets of chromosomes, or 46. In the ovary, meiosis will occur and an egg cell will be produced that has 23 chromosomes. In the male testes, meiosis occurs and a sperm is produced, which will have 23 chromosomes. The gametes are both haploid. Once fertilization occurs, or the sperm unites with the egg, it becomes a diploid zygote. It now has 46 chromosomes. It'll then develop into an adult by mitosis. The alternation of meiosis and fertilization is common to all organisms that reproduce sexually. The three main types of sexual life cycles differ in the timing of meiosis and fertilization. Depending on the type of life cycle, either haploid or diploid cells can divide by mitosis. Now again, only diploid cells can undergo meiosis. In all three life cycles, the halving and the doubling of chromosomes contributes to genetic variation in offspring. That's why sexual reproduction is so important. It allows for genetic variation and eventually evolution. Figure 13.6a is showing the first type of sexual life cycle. This is in an animal, and this is what we saw when we talked about humans. So gametes are the only haploid cells in animals. They're produced by meiosis and undergo no further cell division before fertilization. Gametes, which are haploid, fuse to form a diploid zygote that then divides on mitosis, and we get a multicellular organism. Blue is showing where it's haploid, and pink is showing where it's diploid. There are two other life cycles. We just talked about the animal life cycle. So plants and some algae exhibit an alternation of generations. This is a life cycle that alternates between a multicellular diploid form, this is called a sporophyte, and a multicellular haploid form, called a gametophyte. The diploid organism, which is a sporophyte, makes haploid spores by meiosis. That's how something that's haploid is produced, by meiosis. Now the spores that are produced divide by mitosis. This generates a haploid organism called a gametophyte. A gametophyte makes haploid gametes by mitosis. The fertilization of gametes results in a diploid sporophyte. Figure 13.6b shows the plant life cycle. Now this can be a little bit confusing, so make sure you study this slide carefully so you understand the difference between sporophyte and gametophyte and what meiosis produces and what mitosis produces. The third life cycle, which focuses on fungi and protists, we're not going to look at, and we're not really going to look at plants anymore. We're going to focus on animals. So meiosis reduces the number of chromosome sets from diploid to haploid. And just like mitosis, meiosis is preceded by the replication of chromosomes. Remember, we said before mitosis, there was that interphase, and during interphase, there's the replication of chromosomes. Meiosis takes place in two consecutive cell divisions, meiosis 1 and meiosis 2. These two cell divisions result in 4 daughter cells. So with mitosis, that ended after cytokinesis with 1 division. That's why we had 2 cells as a result of mitosis. In meiosis, there's 2 consecutive cell divisions, which is why we end up with 4 cells, and we call these cells daughter cells. Now these daughter cells only have half as many chromosomes as the parent cell, so the daughter cells are haploid. Figure 13.7 gives an overview of what's happening in meiosis. Then we're going to look at in a little more detail the different stages. We're going to look at how it reduces the number of chromosomes. So first we're starting with interphase, and it's showing a pair of homologous chromosomes in a diploid parent cell. Remember, during interphase is when the chromosomes duplicate or replicate. So now we have 2 pairs of sister chromatids. We have a pair from the father, say in blue, and a pair from the mother in red. During meiosis 1, the homologous chromosomes are going to separate. During meiosis 2, the sister chromatids are going to separate. So that's how, as a result of meiosis 1 and meiosis 2, you're going to end up with 4 daughter cells, each with half the number of chromosomes that you started with. So now we're going to look at the stages of meiosis and cytokinesis. So first we're going to look at meiosis 1, and this occurs in 4 phases. So similar to what we talked about before, when we talked about mitosis, there's prophase 1, metaphase 1, anaphase 1, and telophase 1 and cytokinesis. Now, some books put a prometaphase in between prophase and metaphase, but we're going to follow what your book does, so that it has the 4 stages. It has a PMAP. The first stage of meiosis 1 is prophase 1. And we see 2 unique things happening here that we didn't see happening when we talked about mitosis. So 1 is what we call synapsis. This is when homologous chromosomes pair up. So the pair of number 1 chromatids from your father with the pair of number 1 chromatids from your mother pair up. Then we have what's called crossing over. This is when genetic material is swapped between non-sister chromatids. So one of the chromatids from your father's chromosome number 1 and one from your mother's number 1 are going to exchange genetic material. This is what leads to genetic diversity. It also shows here some other things that are happening. So for example, the nuclear envelope is fragmenting. You can see the mitotic spindle is beginning to form. And you'll see here that it's showing 3 pairs here. Now, if it was humans, we have more than 3 pairs, but it makes it a little cumbersome to show all of them in the cell at once. So they're just showing 3 pairs. The next stage is metaphase 1. So the pairs of homologs line up at the metaphase plate. 1 chromosome, or 1 pair of chromatids, is facing each pole. In mitosis, it was a single chromatid facing each pole. Microtubules from 1 pole are attached to the kinetochore of 1 chromosome of each pair. Now, a key thing to notice here as well is that if you look at the metaphase plate and the chromosomes lined up along the metaphase plate, it's random. In 2 of the 3, the father, which is in blue, is closer to the bottom pole. In the 3rd one, the mother is closer to the bottom pole. So it's random how they line up in the center along that metaphase plate. That also leads to genetic diversity. During anaphase 1, the pairs of homologous chromosomes separate. So 1 chromosome of each pair moves toward the opposite poles. The sister chromatids, however, remain attached to the centromere and move as one towards the poles. Now, if you look at the right-hand side in the upper part of the diagram, you'll see the 3rd chromosome pair moving up. You'll see it has mostly blue but a little bit of red. The one below it is mostly red but a little bit of blue. Again, we're looking at the furthest one on the right-hand side. The reason they have different colors is because during prophase, they exchange genetic material. Finally, we have telophase 1 and cytokinesis. Each half of the cell has a haploid set of chromosomes. Remember, each chromosome still consists of 2 sister chromatids. Cytokinesis usually occurs almost at exactly the same time, just slightly after, and 2 haploid daughter cells are formed. And again, we're talking about animal cells. It's a cleavage furrow that forms between the 2 cells and then it divides. Now, an important thing to know is that no chromosome replication occurs between the end of meiosis 1 and the beginning of meiosis 2. So we don't have an interphase in between the 2. It goes right from meiosis 1 to the process of meiosis 2. And this is just showing all those little individual pictures we looked at so you can see an overview of the whole process of meiosis 1, from prophase through cytokinesis. Now, when we look at meiosis 2, that also occurs in 4 phases, and they have the same name as in meiosis 1, only they're followed by a 2. So you have prophase 2, metaphase 2, anaphase 2, and telophase 2 and cytokinesis. And meiosis 2 is very similar to what happens during mitosis. Figure 13.8b shows the different stages of meiosis 2, and as you can see, they're very similar to what happens during mitosis. So during prophase 2, early on the spindle forms, And later on, the chromosomes are going to start moving back and forth between the two poles. They're going to attach to the microtubules. Then in metaphase 2, the sister chromatids are going to arrange themselves at the metaphase plate. Now, because the crossing over occurred in meiosis 1, the two sister chromatids of each chromosome are no longer genetically identical. Then in anaphase 2, the sister chromatids are going to separate and move toward opposite poles. Finally, in telophase 2, the chromosomes arrive at the opposite poles, nuclei form, and the chromosomes begin to decondense. With cytokinesis, again, that means separation of the cytoplasm. It actually splits into two. So at the end of meiosis, there are 4 daughter cells. Each have a haploid set of chromosomes. Each daughter cell is genetically different from the other cells and from the parent cells. Figure 13.8 is just putting this whole process together. It shows each of those steps we talked about, the 4 in meiosis 1 and the 4 in meiosis 2, just to give you an overall visual of what's occurring. After having gone over meiosis and in the previous chapter, mitosis, we can see that mitosis conserves the number of chromosome sets, producing cells that are genetically identical to the parent cell. Meiosis, on the other hand, reduces the number of chromosome sets from 2 sets, which is diploid, to 1 set, which is haploid, producing cells that differ genetically from each other and from the parent cell. Now, multiple events are unique to meiosis, and these occur in meiosis 1. So we have synapsis and crossing over in prophase 1, and that's when homologous chromosomes physically connect and exchange genetic information. We didn't have anything like that in mitosis. We also have homologous pairs aligning along the metaphase plate. In mitosis, we had sister chromatids aligning at the metaphase plate. And finally, we have separation of homologs during anaphase 1. In mitosis, we had separation of sister chromatids. Figure 13.10a is a visual representation of some of the differences if you were looking at mitosis and meiosis. And we may see a few words that we haven't talked about, like chiasma. Don't worry about those. Just look at the different phases and how physically they look different, what is happening. Now, this summary slide, this table, is a good one to know. This compares mitosis and meiosis. So if we look at DNA replication, it occurs during interphase before mitosis begins. In meiosis, it occurs during interphase before meiosis 1, but not meiosis 2. In terms of the number of divisions, there's 1 in mitosis, there's 2 in meiosis. In mitosis, we do not have synapsis of homologous chromosomes, but in meiosis, we do have synapsis. It's in prophase 1, along with crossing over. As a result of mitosis, there are 2 daughter cells. Each are genetically identical to the parent cell and have the same number of chromosomes. As a result of meiosis, there are 4 haploid cells that are genetically different from the parent cell and from each other. Why is mitosis important? It enables multicellular animals, fungi, or plants to arise from a single cell. It produces cells for growth, repair, and in some species, asexual reproduction. And it produces gametes in the plant gametophyte. Don't get that confused with some of the other things we're talking about, animal gametes. Mitosis is producing gametes in the plant gametophyte only. In terms of meiosis, its role is to produce gametes in animals or spores in fungi and in plant sporophytes. It reduces the number of chromosome sets by half, and it introduces genetic variability among the gametes or spores. Genetic variation produced in sexual life cycles contributes to evolution. So the original source of genetic diversity are mutations. These are changes in the organism's DNA. They create different versions of genes called alleles. So there can be an allele for brown eyes, an allele for green eyes, an allele for blue eyes. The reshuffling of alleles during sexual reproduction produces genetic variation. The behavior of chromosomes during meiosis and fertilization is responsible for most of the variation that arises in each generation. There are three mechanisms that we're going to look at briefly that contribute to genetic variation. And these we've talked about already a little bit. The independent assortment of chromosomes, crossing over, and random fertilization. So first, the independent assortment of chromosomes. So homologous pairs of chromosomes, if you remember when I showed you that slide earlier, when you're looking at meiosis 1, they orient randomly at metaphase 1 of meiosis. Each pair of chromosomes sorts maternal and paternal homologs into daughter cells independently of the other pairs. The number of combinations possible when chromosomes assort independently into gametes is 2 to the n power. So for humans, n, and n, if you remember, represents the number of chromosomes in a set, equals 23. So for humans, there are more than 2 to the 23rd power possible combinations of chromosomes. This means it's over 8 million possible combinations of chromosomes. Figure 1311 shows independent assortment of homologous chromosomes in meiosis. So say we're only looking at 2 pairs of chromosomes, one set from the mother and one set from the father. On the left-hand side is one possible arrangement. You can have 2 from the one sex on the left-hand side of the metaphase plate and 2 from the other sex on the right-hand side of the metaphase plate. When that cell splits in half, the blue ones from the one parent are going to the cell on the left, and the red ones from the other parent are going to the cell on the right. The other possibility is what we see in possibility 2, where we have one pair of chromatids from one parent and one pair of chromatids from the other parent on the same side of the metaphase plate. So as you see, when that cell divides, you're going to have one pair from the father and one pair from the mother in each of the new cells. So that's what we're talking about. We're talking about independent assortment. They line up randomly on that metaphase plate. Crossing over is that second mechanism that we talked about. This produces recombinant chromosomes. They have DNA from 2 parents in a single chromosome. And in humans, an average of 1 to 3 crossover events occurs per chromosome. So this is showing how crossing over can cause genetic diversity. So if you see, it's showing it in steps 1 and 2, how you have chromosome material exchanging between non-sister chromatids. One of the chromatids from your dad and one from your mom are exchanging genetic material. That's why when we see in number 3 and below, some of them aren't fully one color. They have a mixture of blue and red. That's because they've exchanged genetic material. Now, they didn't show that in the slide with law of independent assortment because they just wanted to highlight the law of independent assortment. But you would see that on there as well. And finally, random fertilization. This adds to genetic variation because any sperm can fuse with any ovum, any unfertilized egg. The fusion of 2 gametes produces a zygote, and there's about 70 million different possible diploid combinations. That's why everyone is different from everyone. Now, what's the evolutionary significance of genetic variation with populations? So natural selection results in the accumulation of genetic variations favored by the environment. Sexual reproduction contributes to the genetic variation in the population, and this occurs from mutations. In general, animals that always reproduce asexually are quite rare. One example is the bdelloid rotifer. And what they do, they increase their genetic diversity through the incorporation of foreign DNA from the environment. And this is just showing a picture of an animal that reproduces only asexually, that rotifer. And like I said, it's very rare for an organism to only reproduce, for an animal only to reproduce asexually.